I know some of you, most of you maybe. I'm Joe Wong. I'm currently at the San Francisco VA and UCSF. Been there about 15 years. I met Doug in 1992 when I came to do my ID fellowship. I was, of course, deeply in awe of him. At that time, Doug had already waded into the field of HIV therapeutics and was a pioneer in so many ways. In addition, he had been the first to describe HIV drug resistance, and not only that, but really identify resistance, genetic resistance testing as a way of being able to characterize the virus, and then also as a clinical tool. And to this day, 30 years later, these observations that Doug made, along with Brendan Larder and Sharon Kemp and others, are still sort of the bedrock of what we do in managing our patients. And so, again, you know, anyone who knows Doug knows that he's such a kind and approachable person. But when I met him, I knew of his reputation academically, and I was totally in awe of him. I was quite surprised that he was interested in having me consider rotating with him after I finished my clinical rotation. But my final decision to work with Doug, actually I have to thank for another senior ID faculty member. I was on rounds at Hillcrest with Elizabeth Ziegler, and some of you probably remember Dr. Ziegler as a very petite but imposing senior faculty member. At least when I was on rounds with her, she didn't smile very much. But I think it was probably 8 or 9 o'clock at night, and we were just waiting probably to review some radiology with the radiologist. And she asked me what I was interested in doing in terms of research. And I'd mentioned, well, HIV would be something interesting. What should I do? And she turned to me and said, well, if you're interested in HIV, you've got to work with Doug Richman. So that kind of really fixed it for me. And then in addition to this picture of Doug, who of course is ageless, I don't think there's much of a difference looking at that picture to seeing him in person now. I put some pictures of other people, some pictures that you've already seen. One of the things that was really striking as I came to the lab and got to know people was just the amazing group of people that Doug had surrounded him with across the board, from senior scientists like Chelsea and Richard Kornbluth to junior scientists at that time like John Guatelli. And then when Topher showed his picture of that postdoc room, well, I was in that postdoc room at one time, and the people that were with me were Susan Little, Pascal Melan, and Michal Chowers, another fantastic group of people. And so it was very easy to feel welcome. And I feel so privileged to have had a chance to train with Doug and to work with Doug these many years. So that's sort of my bit as far as how I met Doug and decided to work with him. The one amusing anecdote that I would like to share with you is, I can't remember exactly what year, but probably around 2000, 1999, something like that. And Doug, as you guys know, has been an organizer of one of the international AIDS conferences, the Conference on Retroviruses and Opportunistic Infection, called CROI, for many, many years. And one of his favorite parts of being in CROI is not to sit in a lecture hall and listen to lectures, but to go to the poster sessions, because it allows you to interact with young scientists, to look at data and discuss data in ways that can't be done in a big lecture hall. And I am still in awe of Doug and his ability to quickly grasp what's going on in a poster hall of 500 or 1,000 posters, and his passion and enthusiasm for science as he would flit around from poster to poster and drag some of us to also partake in some of those posters. So that's my anecdote. And then I also wanted to share a couple of the Dougisms that I remember. So that first one, the perfect, is the enemy of the good. I think for some of us like Satish and me who are natural procrastinators, I think that was his way of trying to encourage us to go ahead and stick our necks out and publish and the work that we've done. The second, I think, actually has many layers of meaning. The data are the data. So there are times as scientists where we are disappointed with the things that we find. And Doug was never that way. I think he felt that if you design an experiment well and do it well, then the data have to speak for themselves in terms of what they tell us. And then on occasion, even when the data don't tell us something that we expect, they can teach us really, really important things. And then the last thing I think is again something that, and it's a paraphrase, I don't know exactly, I can't remember exactly Doug's words, but it's really important I think to enjoy the work that you do. And I think Doug really created an environment in his lab group that really made it possible for us to really enjoy doing our work. So my title was a little bit curious probably, and for those of you who are Tolkien aficionados will know that that title, my title, was actually lifted from the title of the journal that Bilbo Baggins started in The Hobbit and that was finished by Frodo Baggins in The Lord of the Rings. And so I'm going to use this as sort of a setup for how I perceive the last 30 years of the HIV epidemic. So when I arrived in San Diego, we had very limited options for treating patients. And HIV was really a devastating disease, and things were very, very bleak. But into that breach, people like Doug and Alan were really heroes to figure out how we can manage patients and care for them and to develop the tools that would allow us to conquer the disease. So, by the mid-1990s, again, thanks to the good work that Doug and others have done, it looked like we were ahead of the disease. This is the end of The Hobbit and the Tolkien story. Lots of health benefits, and this graph is really sort of a composite of several graphs, including the work from Frank Palella showing the decline in mortality in HIV patients with the advent of combination therapy. And in fact, the benefits seemed so remarkable that we were really sort of hopeful after many, many years of bleakness that we were on the cusp of maybe eradicating HIV as a disease. That turned out not to be the case, but because Doug had been involved in one of these seminal studies, the Merck 035 study that again had been one of the first to really introduce protease inhibitors in combination with nucleoside reverse transcriptase inhibitors into treatment, we were poised to be able to look at what remained in patients that had been treated with these drugs for a while. And I think, again, these studies were made possible in part because of fantastic people like Huldrych and Chelsea and Marjan and Diane Havlier and other people that were working here with us. And so our group was one of the three to really report that in spite of really good suppression of the virus, that some reservoir of latent infection remained. The picture here is of the Tower Barad-dûr in the land of Mordor, which is the focus of the 3 books of the Lord of the Rings trilogy, and where the Dark Lord still lurked in spite of the good conclusions in The Hobbit. On the right, the graphs are from the Silicano group, showing really sort of the stability of this reservoir over very many years using quantitative viral outgrowth assays. So the consequences of persistence of latent reservoir were many. We know that treatment interruption results in return of viral replication, necessitating really lifelong therapy. And then, as others have already summarized, we know that viral persistence likely gives rise to persistent immunopathology, drives a lot of non-AIDS morbidity that still plague our patients, especially those that are started on treatment relatively late after infection. In 2008, I think, at Croy, I think there was a report that there was a patient that might have been cured, published in the New England Journal in 2009. This is Timothy Brown, the Berlin patient. And this generated a lot of excitement that, in fact, after 10 years of trying to study the latent reservoir and how to get rid of it, that this could be achieved. This patient, of course, had undergone quite a bit. He had been diagnosed with leukemia, underwent 2 consecutive allogeneic stem cell transplants, and got transplanted with cells from a donor who happened to be delta 32 homozygous at the CCR5 allele, so that the donor cells were essentially resistant to HIV infection. Apparently now, after another decade of follow-up, he remains off therapy and has not had viral rebound. Really, the first example of an individual with cure of HIV. A second patient, a London patient, might have been described earlier this year, also having to undergo stem cell transplant. So this is my final tie-in with the Lord of the Rings. And there you see Doug leading one of the merry bands of intrepid heroes in the quest for a cure. I'm somewhere in the middle there. The tall guy is probably Hildrick, and the guy at the end is probably either Davey or Satish or someone like that. You also notice that what I'm carrying looks kind of like a lacrosse stick rather than an axe, which explains why I haven't been more successful. I've in a different way showing a slide that some of you guys have already shown, just sort of categorizing strategies for effecting a cure. The nuclear option is something that could basically wipe out all the potential CD4 T-cells in a patient, like stem cell transplants and lots of X-ray therapy and other things. Having said that, though, we know that, in fact, the majority of people who undergo stem cell transplant who have HIV actually aren't cured, and that there are many cases where people have undergone stem cell transplant and actually had rebound sometimes later on. The second is the shock and kill strategy that Doug, in a nice review in 2009, really kind of laid out. This is the idea of trying to induce viral expression in order to trigger viral cytotoxicity or elicit immune clearance. The remainder of my talk and the data part of my talk will really concentrate on the shock and trying to characterize what is relevant in terms of viral expression and what can be accomplished with the tools that we currently have. And then a number of other things that Satish and others have talked about, sort of more viral specific detection and curing or killing. I've sort of just listed very briefly there. So again, this another review from Steve Deeks with a cartoon that you guys have seen before. The idea being that we can do something to force virus to be expressed from a latently infected cell so that it's no longer latent, and then either through immune recognition or cytotoxic effects from products from the virus, the cell dies. So this summarizes some of the data on one class of these drugs. This is the paper from Nancy Archin and Dave Margolis. Doug and Matt were involved in these studies. The white bars show the baseline levels of cell-associated viral RNA, and the dark bars show the amount of viral RNA that can be induced in them after treating with a single dose of vorinostat in vivo. And what you can see is that on average, something like 4 or 5 times as much RNA can be induced to be expressed in these cells. So it was a proof of concept that in vivo you could actually cause these cells to kind of come out of hiding to some degree. However, a number of studies looking at vorinostat as well as other newer HDAC inhibitors, despite showing a similar ability to cause an increase in viral expression, don't seem to have any measurable effect on the size of the latent reservoir. So we were interested in this, and we wanted to think about what constitutes the transcriptional profile of latently infected cells, and then also what might happen when you treat these cells, at least ex vivo, with drugs that have the potential to reverse latency. And these studies really still draw a lot on some of the principles that Doug initially conveyed to me. One of my first studies actually was to look at HIV drug resistance patterns in tissues and specifically in brain. It was this idea that, in fact, if you look just at blood, that you might miss things that might be going on that are informative about pathogenesis and other things. And so we wanted to, in this case, look at GI tissues, also GU tissues, because other studies from the UCSD group, including a lot of work from Davey, show that the GU tract can also function as a reservoir for virus. So the studies that I'm going to show you depend on this transcriptional profiling approach that had been developed with Philip Kaiser and Steve Uckel. And one of the main innovations here is that they developed a way of actually measuring transcripts that have been initiated but not elongated. I won't go into detail about that, but because the TAR loop forms a stem loop, and if that falls off without elongation, it's actually a very tight stem loop that's hard to actually reverse transcribe. And so a lot of conventional RT-PCR assays actually won't pick it up. So they had developed a method using methods from microRNA reverse transcription to tackle that problem. And then a number of the other assays actually have been adapted from observations and reports from others. The read-through assay based on work from Mattia Peterlin and Bob Silicano's groups, and then also the poly-A assay, which the Silicano group feels is probably the closest in terms of a transcript to give you an idea whether a cell that's infected with HIV can make protein. All HIV RNA should have, transcripts should have the TAR loop. If they get beyond the stem loop, they should have the long LTR product. And then as they go down, they'll have POL, which is really the equivalent of a GAG product, NEF, the poly-A, and then we know that multiply spliced RNA, as Marta talked about, the tilde assay seems to be a marker maybe of productive infection. And so this basic set of 5 to 7 assays were applied in a number of different settings. So this is the original paper from Steve Uckel looking at peripheral blood CD4 cells. And what you can see is so in general, one of the surprising things that Steve found is, again, these are patients who've been well suppressed, no detectable viral load. I think the median may be something in the order of 7 years of suppressive therapy, that in spite of being arguably latent cells, these cells actually show a lot of TAR message, on average of 20 TAR copies per provirus. And so there's a lot of initiation going on in these cells. Most of this is not accounted for by read-through. So read-through would be transcription through a gene that the provirus is integrated in. So mostly not explained by read-through. And then there is a drop-off in terms of elongation, about a log, lower elongated, proximal elongated products. I'm sorry, here proximal elongated products. And then in terms of RNAs that become completed or spliced, much lower. This pattern really sort of follows, this was normalized to RNA, this is normalized to actually proviruses, and so again, that pattern really seems to be consistent. The apparent block to elongation seems to be not restricted from the TAR to Long LTR. When they added assays for Pol and NEF, you can see really sort of a gradient where there are fewer of the Long LTRs than the TAR loops, and fewer yet of the Pol, the NEF, and then finally poly-A and TAP-REV. One question that could come up, because we know that there are lots of mutations that could potentially affect the sensitivity of these assays, is whether variations in sequence might be affecting our estimates of either individual assays or of the DNA. And so in this particular plot, the data are actually the RNA assays normalized to the same assays for DNA. And this gradient persists. So those data really show that, again, even though we think about latent infection as being quiescent, they're not truly quiescent, at least in, as these data show us, that there really is a lot of transcriptional initiation, and then something happens to prevent the cells from making these longer transcripts. The other issue that comes up, and again, thinking back to the questions that we approached early in our studies of patients on long-term therapy, is how much of the T-cell reservoir is in the peripheral blood compared to every place else. And this is an approximate that roughly 2% of the T lymphocytes in the body are in the circulating blood. A majority of the cells probably are in the gut-associated lymphoid tissue, and then everything else, other mucosal sites, central secondary lymphoid tissues carry the rest. And so we were interested in profiling this transcriptional pattern to see whether it would be similar in these other sites. So this is data from Sushama Talwat, where she looked at PBMC in gut. Again, initially just normalized to the cell numbers. So that is easy for PBMC in gut, because intact gut tissue, we can't say what the CD4 content is. We'll talk about it in a little bit. But the overall pattern is not dissimilar. So I'm sorry, this is PBMC, so it's not going to be dissimilar. So again, similar to what I showed earlier, low levels of transcriptional interference, high levels of transcription initiation, and then strong blocks to either proximal elongation, and then additional elongation and completion and splicing. So this is now intact gut biopsies that she looked at. And the idea of using the intact gut biopsy, this is the closest that we can get to what might be present in vivo. Again, in the gut, she also found that there's relatively little read-through to explain the relatively high TAR. And again, a gradient from initiated transcripts down. The blocks to elongation, completion, and multiple splicing in the PBMC also seem to be present in the intact gut tissues. However, she also, in some of the samples, was able to get sufficient material that she could actually dissociate the CD4 cells and then analyze the isolated CD4 cells separately from, again, the stromal tissue. And here, the pattern actually is a little bit different. So in terms of proximal elongation, again, if you look here, the darker colors are the intact rectal tissues. You see, again, the step off between initiated transcripts to ones that are elongated and so forth. But in the isolated cells, interestingly, that block seems to have been released. And this difference seems actually to be robust and reproducible, and the additional observations that were noted was that the read-through transcripts in the rectal tissues were even lower than in PBMC, and then the total TAR were somewhat lower than in PBMC, and NEF were again slightly lower than in PBMC. In the intact tissue, again, the normalization here is for provirus. So again, each infected cell, how many transcripts are seen, and it looks like somewhat surprisingly in gut, even though we think of it as sort of an activating environment with lots of microbial products and other things present, doesn't seem like it actually stimulates a lot of viral production compared to PBMC. And then in the isolated cells, there's the suggestion that whatever this proximal elongation block was, it seems like it may be actually context specific, that it has to be present with stromal cells and other things. Alternatively, I think you could also say that when you isolate these CD4 cells from the gut tissue, that you've done something to them that may now be a departure from what's present in vivo. For the first hypothesis, I think that there's actually some possibility, because we know that in stroma there are actually a lot of T regulatory cells that probably create a negative signal for cells, and perhaps in intact tissues you see this block that might be absent once you've isolated the cells away from those inhibitory cells. So there were blocks to completion of HIV transcription and splicing in both gut and blood. Block to proximal elongation was observed in rectal biopsies in blood, but not in rectal CD4 cells. There was a greater block to HIV transcriptional initiation in the rectum, both in the rectal biopsies and in sorted CD4 T-cells compared to the blood. And again, that seems a little bit surprising given the higher proportion of T-cells in the gut that carry classic activation markers. And finally, that the slower HIV transcription in the gut could reflect differences from blood, either due to integration sites, epigenetics, levels of cellular transcription factors, and or again, effects of T-cell activation. So the final, actually this is not the final, next to final set of data are just a smattering of data showing how LRA treatment can reverse latency, comparing again blood to gut, same patients. So this first slide shows how well anti-CD3, anti-CD28 treatment can reverse latency. Treatment either in PBMC or in rectal cells really seems not to affect read-through at all. In contrast, I'm sorry. So the gray is the DMSO control, and red is treatment. So when you treat with anti-CD3, CD28 in PBMC, you see an increase in initiation, increase in proximal elongation, increase in completion, and increase in multiple splicing. In rectum, there's a minor increase in read-through, but healthy increases in initiation, elongation, a big increase in completion, and splicing. In contrast, if you look at romidepsin, one of the stronger HDAC inhibitors, we see significant increases in initiation, elongation, and completion, but not in this increase in splicing, and that's the case in both PBMC and rectum. So those data, she's done additional analysis of other HDAC inhibitors and PKC agonists, and they basically do show that in fact the reservoir cells in gut seem to be inducible. So the last bit of data I want to show are some data now that include blood, gut, and genital tract, relatively few cases that we actually got liver. And this was a collaboration with the Weiss cohort led by Phyllis Tian at UCSF. The numbers here are relatively small, 6 patients, again, cryopreserved PBMC, fresh frozen biopsies from ileum, colon, rectum, and then from the genital tract, 3 sources of tissue, cervix, endocervical curettage, and endometrium. These were all collected from premenopausal women in mid-cycle. Relatively few liver samples. So this is just an analysis of the DNA content, either total DNA or a measure of intact DNA using an adaptation of the Silicano intact DNA, intact provirus DNA assays. Again, when this is normalized to millions of cells, realize that the level of CD4 could vary between them. And so even though the HIV DNA copies per million total tissue, PBMC cells is the highest, this has not been corrected for CD4 content. Nevertheless, it's interesting to note that if you look at the ratio of the total DNA to the intact proviral DNA in blood, the difference is about a log, which is in keeping with what has been reported. But in the tissues, this delta seems to be actually much greater, and it's really between 1.5 to 2 logs. So for whatever reasons, it looks like in this small sample that we have, that there may be more defectives present in these tissue samples than in the peripheral blood. And then looking at whether this the provirus is inducible, again now here described as HIV RNA to HIV DNA, so no longer dependent on this variability in terms of CD4 count. What Sarah Moran Lopez found was that, here, as we had previously shown in other patients, that there's a lot of transcriptional initiation, less proximal elongation, less polyadenylation, and very low levels of multiple splicing. But the levels per provirus of initiation certainly in gut, less so in endometrium, seems to be considerably lower than in peripheral blood. And then there was still sort of a comparable decrement in terms of proximal elongation, completion, and splicing. One of the interesting things is that within the female genital compartment, the endometrium seemed really different than either the cervix or the endocervical lavage. And one of the potential explanations and one of the challenges in studying these tissues is the possibility that there's confounding from contaminating blood. And so Sarah came up with a cool way of looking at this in a collaboration with Nadia Rohn's lab. And so they basically did cytoph on the CD4s that were isolated from the cells from these different compartments. And this N of 1 data here shows these TSNE, two-dimensional renderings of the cytoph multidimensional data. And what you can see is that blood has this pattern here, and then ileum, colon, and rectosigmoid here, and then endometrium here, really probably overlapping with some of these, although I think if you did a TSNE between these, they would probably be different, but clearly not overlapping with blood. And so this similarity in terms of the transcriptional profile in the endometrium seems to really not be because of contaminating blood, because the T cells that were isolated from those endometria seem to really be distinct from what was found in blood. So quick summary, not only reservoir size, but HIV transcriptional patterns differ between blood, gut, and different female genital tissues. The latency reversing agents variably increase initiation, elongation, completion according to tissue source. And then only anti-CD3, anti-CD28 co-stimulation in the studies that we've done seems to result in significant increases in multi-splice TAT-REV RNA. And then there's this higher ratio of total HIV DNA to the intact proviral DNA values in mucosal tissues we found it in blood. Small sample size, but sort of intriguing that maybe a lot of these proviruses that we're measuring in the tissues, in these mucosal tissues, may actually not even be as relevant. So my last is, again, a thank you to Doug for, again, the many years that he supported and encouraged my work. The Dirty Dozen old-timers that I had the great pleasure of working with and still work with to some degree. Folks now that I work with at UCSF, the data were generated primarily by Sushama and Sarah, and under Steve Ukel's leadership. Other folks in the lab, including some people who have left, who were instrumental in developing some of the assays that we're using, collaborators at different sites at UCSF, and then, of course, the dedicated patients and research participants across the many sites and over many years, and our funders. Thanks.